In our previous lecture, we spoke about the cut and paste mechanism of transposing. But this is not the only technique that we get. We also have a copy and paste mechanism, and this is associated with retrotransposing. What we want to understand in this mini lecture is actually how do we perform this copy and paste mechanism. With a copy and paste mechanism, if you look at the illustration that I've presented somewhere here in the note, is that we're moving from a double-stranded DNA to an RNA into a cDNA, then the cDNA would eventually make a complementary strand, which is going to become a double-stranded DNA. So that's how we move from this copy and paste mechanism. The example that we're presenting here is the L1 element, which is a retrotransposon. So it performed this activity with the help of two protein, ORF1P and ORF2P. So from this picture here, as you're going to see, is that the L1 element, the DNA, is going to become first an L1 RNA, and this L1 RNA is going to actually produce those two protein, ORF1P and ORF2P. And those ORF1P and ORF2P are actually endonucleases that are going to create a nick in the DNA of the next destination. And once there's that nick, there's a free flanking 3' hydroxyl group, and that flanking hydroxyl group is actually going to allow the synthesis of a cDNA from that RNA, as you see. And as soon as we've got the cDNA, eventually we're going to have a complementary strand and a DNA repair mechanism is going to occur. So that's how, in a nutshell, we have the copy and paste mechanism in retrotransposing.